Hello, hello. We are picking up with the respiratory system in the trachea. We're going to make our way from there. So, trachea, of course, is our air tube. It is going to run from the larynx, which we see right here. There's the thyroid and cricoid cartilage. And the trachea runs down until it splits in your chest into these main bronchi. So it is our airway. It has conducting zone functions, which means it is going to be warming that air, humidifying that air, and filtering that air as it moves it. Now, most of the trachea, if we slice it, has this ring that's more like a C. It's not like a complete ring, but this C-shaped ring of hyaline cartilage, and that gives the trachea structural support and primarily prevents it from collapsing. Now notice that the cartilage is absent in the back, and instead in the back here we have this muscle called the trachealis muscle. It's a smooth muscle, and you're going to use that muscle when you are like coughing or something. But besides having that nice muscle here, the absence of the cartilage allows the tube behind it, and the tube behind it here is the esophagus, allows that esophagus to expand when you're swallowing a big bolus of food, like a big mass of food and saliva all smooshed together. Now, the trachea has an adventitia around it. The adventitia is going to anchor it. Particularly, it's going to anchor it to the adventitia of the esophagus, which is right next door. Meanwhile, the trachea has a mucosa. We see cilia here. There would be mucus glands in here, plus goblet cells to make mucus to help do that filtering and humidifying. The cilia, of course, are going to sweep upwards. So you're constantly sweeping mucus with, dust in it, bacteria in it, viruses in it, constantly sweeping it upwards, and you're always unconsciously swallowing it. All right, fantastic. Let's look at some histology. Let's look at some histology. There we go. So here I got my trachea's lumen over here. Here's the epithelium, which is going to be pseudostratified columnar epithelium. I do see some little goblet cells, like that little guy right there. That little guy right there, those are goblet cells. We know goblet cells make mucus. And I can totally see the beautiful cilia, which are always sweeping the mucus upward. Underneath that mucosa, we got a submucosa. This is mostly connective tissue. Blood vessels in here too. Then outside that, this is the beautiful hyaline cartilage, which is going to prevent that trachea from collapsing. All right, from the trachea, we go to the lungs. Look at the lungs. Look how they occupy that thoracic cavity. Notice all these ribs protecting the lungs on each side, the scapula, the sternum, the costal cartilage as well. Look how the apex of the lung is actually sitting above the clavicle, like up into your neck, both sides there. Notice how the base of the lung, which is down here, is going to rest on this diaphragm. We see this dome-shaped, relaxed diaphragm right there. If we take the lungs out and take a look at them, we got our right lung over here. Apex, of course. Base at the bottom. Notice that the right lung has 1, 2, 3 lobes. A superior lobe, a middle lobe, and an inferior lobe. The superior and middle lobe are separated by a deep indentation called the horizontal fissure. Meanwhile, the middle and inferior lobe on the right are separated by a more angled indentation called the oblique fissure. Oblique means at an angle. Now, if we contrast this with the left lung is smaller because the heart takes up some space. We see what's called the cardiac notch indentation for that heart. And note there are only two lobes on the right-hand side, left-hand side here. There's a superior lobe and an inferior lobe, and they are separated by an oblique fissure. Notice an oblique fissure is always next to the inferior lobe, both sides. If we look at the medial surface of the lung, this is the right lung. We can still see its nice apex up here. We can see its base down here. We got the three lobes, boom, boom, boom. We got that fissure right there, that horizontal, oblique. Now what we can also see so nicely is the area where everything goes in and out. It's an indented region called the hilum. And going into my lung, I have a big air tube called my main bronchus. That's a branch from my trachea. So remember, my trachea comes down, my trachea branches into one branch for each lung, and that big branch is called the main bronchus. Now, also going into my lung, I have a pulmonary artery. Notice it is a weird artery. It is blue because it is deoxygenated. Then I got two pulmonary veins coming out. They're weird veins because they are oxygenated. So we got some good lung anatomy there. We switch sides. It's the same exact idea. Again, my apex, my base. Remember, only two lobes on the left, inferior, superior, and an oblique fissure. There is an indentation for the heart. There's a little notch in the lung called the cardiac notch. Then like a region where the heart would smush into it called the cardiac impression. Another hilum, main bronchus going in, weird pulmonary artery going in, weird pulmonary veins coming out. So we got some good, like, basic fundamental lung anatomy there. And if we consider the branches in our lung, like the airway tubes, the big ones, those are called bronchi. So my trachea comes down, my trachea splits into a right and left main bronchus. Each lung gets its own main bronchus that goes in and at the hilum. Now, the main bronchus is going to separate and give us lobar bronchi. So every lobe gets a lobar bronchi. So my right lung has 1, 2, 3 lobes. So my right lung will have 3 lobar bronchi. My left lung will have 2 lobar bronchi. Then my lobar bronchi will split to give me what are called segmental bronchi. Then those will split to give me smaller and smaller bronchi. So we got all this splitting going on here. And one of the big effects of this is going to increase surface area. Because we want a big surface area for exchange. If we look at a color-coded picture here, we got our trachea at the top, coming down and splitting. In green, we got our 2 main bronchi, one for each lung. Notice the left one is longer because it's got to get over the heart that would be sitting here. Now my main bronchi split to give me lobar bronchi. 1, 2, 3 for the right, 1, 2 for the left. Then lobar bronchi split to give me segmental bronchi in the yellow, and they split to give me even more bronchi and even more bronchi. This is sometimes called the bronchial tree because of the way the branching is taking place. All right, continuing and kind of reiterating, we got our trachea. The trachea is going to split to give us our main bronchi. Here's the left one. The left main bronchi is splitting to give us my lobar bronchi. Lobar bronchi is splitting to give segmental bronchi. Segmental bronchi is splitting to give smaller and smaller bronchi. Now what I want you to notice first of all is the cartilage. Remember we had the cartilage in the trachea? And the purpose there was to prevent collapse. Same idea in these bronchi. But as we branch, the danger of collapse is going to get lower and lower and lower. And because of that, the amount of cartilage is going to get smaller and smaller and smaller. A small tube is in less danger of collapse, so it doesn't need the cartilage. Plus, cartilage would get in the way of exchange. So we got to kind of gradually get rid of that cartilage as we do our branching. Now eventually we get tubes without cartilage. A tube without cartilage is called a bronchiole. And bronchioles will branch. Bronchioles are pretty cool. They don't have cartilage, but they got muscle. Look at their muscle right there. And that muscle means they can constrict or dilate to let more or less air in. Now eventually we get bronchioles with alveoli. See that little ball right there? That little guy right there? Those are alveoli where gas exchange happens. And when I have a bronchiole with alveoli, that's called a respiratory bronchiole. And the last bronchiole without alveoli is called a terminal bronchiole. And then eventually we get these big just sacs of alveoli. And of course, where we have alveoli, that's our respiratory or exchange zone that we're in. Okay, knocking out some good anatomy of the lungs here. Got some histology. These are a pair of bronchi. This is the lumen of a bronchus right here. This is the lumen of a bronchus right here. I can tell they're bronchi because I see this cartilage plate here, this cartilage plate here, here, and here. This is a bronchiole up here. It doesn't have any cartilage in its wall. I also see little alveolar sacs. All these little indentations, right there, right there. Those are alveoli. So nice view of the lungs here. Now, as we said a moment ago, bronchioles are going to lack cartilage. Now, generally, they have a diameter of less than a millimeter. So, there are two named bronchioles that we're interested in. Respiratory bronchioles are bronchioles with some scattered alveoli. A terminal bronchiole is the last bronchiole without alveoli. So a terminal bronchiole, terminal means at the end. Well, the end of what? The end of the conducting zone. Now, our respiratory bronchiole will turn into an alveolar duct, which is a tube made of mostly alveoli. Then that'll dead end. And that dead end of alveoli is called an alveolar sac. All right. So as we were saying a moment ago, when we get our respiratory bronchioles, we start seeing alveoli. That means we are in the respiratory or exchange zone. Respiratory bronchioles begin this zone. They have some alveoli, and they have a simple cuboidal epithelium in between. Respiratory bronchioles give rise to alveolar ducts. Like this is an alveolar duct where my arrow is right now. That's a tube made predominantly of alveoli. Alveolar ducts turn into alveolar sacs, which, as we said a moment ago, are dead ends made of alveoli. Here I have a bronchiole right here. This is a terminal bronchiole over here. And then he's becoming a respiratory bronchiole and then becoming an alveolar duct. Like, look at those alveoli right there. Boom, 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 boom. And eventually that duct would end in a sac. Now our alveoli, and here's one right here. They are my sites of gas exchange. We have about 300 million of these, and they're made of simple squamous epithelium. So here are a bunch of alveoli right here. They're in an alveolar sac. Simple squamous epithelium is right there. Notice the little pores connecting the alveoli to one another. Notice that they're covered in blood vessels. Those are pulmonary capillaries, which are, of course, crucial for exchange. Notice also that my alveoli are covered in elastic fibers. This is really important. What this means is that when you breathe in, you stretch your alveoli, they expand, right? But then because they're covered in elastic fibers, those fibers want to recoil. So when you're done breathing in, the fibers recoil and snap back, and that is going to help you breathe out. Smoking can destroy those elastic fibers, making it harder for you to breathe out. So don't smoke. All right, well, going back for a second. What I want to do is zoom in on this, where I drew a little square right here. I'm going to zoom in on a couple side-by-side alveoli. And when I do that, like this is the interior of an alveolus right here. This is the interior of another one, a third one, a fourth one, a fifth one. I see little pores letting air flow between them. My alveolus is made of a simple squamous epithelial lining, and I got a simple squamous epithelial cell I'm on right here. In fact, this whole guy right here, this whole guy right here, these are simple squamous epithelial cells. The simple squamous epithelial cells are called alveolar type 1 cells. They are where gas exchange occurs. Look how there are next door pulmonary capillaries to pick up oxygen and get rid of CO2. Now, my simple squamous cells are type 1 alveolar cells. They're also type 2 alveolar cells. We'll talk about them in a moment or so, okay? There are also alveolar macrophages that wander around your alveolar surfaces looking for, particles like dust particles, looking for bacteria, viruses, those sort of things. All right, now what I want to do is zoom in on this little area with my type 1 simple squamous epithelial cell and also the simple squamous epithelial cell of my capillary. So I'm going to do that. There we go. I zoomed in on them. Here's my alveolar air. Here's my simple squamous type 1 alveolar cell here. Here's my capillary simple squamous cell. Here's my blood. Here's a red blood cell. Oxygen will diffuse from the air in the alveolus into my blood and into my red blood cell. CO2 will diffuse the other direction from my red blood cell, from my blood, into my air. The combo of these two epithelia, as well as a little bit of proteins in between, is known as the respiratory membrane. And that is your exchange surface. All right, so we're now zooming out to talk about one more anatomical feature of the lungs, and that is the double-layered serosa, the double-layered membrane covering each lung. Each lung has a pair of membranes around it. There is an outer one called the parietal pleura and an inner one called the visceral pleura. The visceral pleura is on the lung itself. The parietal pleura will be on the inside of your rib cage, on the top of your diaphragm, that sort of thing. Now there's a tiny space between those two pleural membranes, and that tiny space is called the pleural cavity. It is filled with pleural fluid, and one of the jobs of that fluid is going to be for lubrication. And we did a lab 2 and A&P1 about this. And the lubrication is necessary because the lungs are, expanding as they inflate and then getting smaller as they deflate. All right. If I zoom in, I got some lung air. This is alveolar cells right here. There's alveoli in here. And then I've got my pleural membrane right here. I got my visceral pleura right there, which is kind of cool. If you've ever eaten ribs, you can see the parietal pleura on the inside of like a slab of ribs, like shiny plasticky looking substance. So I just told you that the pleural fluid, which is in the pleural cavity, which is the space between the pleural membranes, I just told you it was there for lubrication, but there is another function of it. The other function of it is going to couple, like join, the motion of the thoracic wall to motion of the lungs. All right, let's do a little experiment here. Imagine you had a glass table and a glass of water, and you pour some water onto the table. Then you stick the glass right onto that spilled water. The water will make the glass stick to the glass. Try this at home if you don't believe me. Or go to like a store that has a glass table and a glass and do the experiment there, right in the store. And what this means is that because of these two membranes and the fluid in between, when you move your ribs or move your diaphragm, the fluid makes the lungs move with it. So when your thorax gets bigger, your lungs get bigger. And this is really important. We got to make sure when the diaphragm moves, the lung moves. When the ribs move, the lungs move. And the pleural fluid and the pleural membranes help me do that. All right. So now we're going to get into the mechanics of breathing. And to understand the mechanics of breathing, we got to be aware of three important pressures. Number one, there is atmospheric pressure. That is the pressure that air is exerting like around you. Number two, there is the pressure in your lungs. That's called intrapulmonary pressure. Number three, the pressure of your pleural cavity. That's intrapleural pressure. So three major pressures. Of course, you don't affect atmospheric at all, but you can alter these two. Now, what I want you to notice, first of all, notice intrapleural pressure, the pressure in the pleural cavity, is less than intrapulmonary. That always has to be the case. Intrapleural always has to be less than intrapulmonary. We'll see why in a second. Here's why. This came quicker than I thought. Here's why. Remember, the lungs are surrounded by what kind of fibers? Elastic fibers. And so when there is air in your lungs, your lungs are stretched. So your lungs are stretched. What that means is that the wall of the lungs is wanting to come back inward. The walls of the lungs, because they're stretched, wants to come back inward. So your lungs want to collapse because of that elasticity. However, pressure is higher in here where I'm putting some blue dots. Pressure is higher in here and lower in the pulmonary cavity. So air is always trying to go from the lungs into the pleural cavity. And that is going to resist the elastic tendency of the lungs to recoil. So we need this intrapleural pressure to always be lower than this intrapulmonary pressure, or else the lungs will collapse. Now let's suppose that a student accidentally stabbed me in class with a little knife. And when they did so, all of a sudden air got into my pleural cavity. So in this situation, the pressure of the pleural cavity, so the pressure of my pleural cavity, there we go, the pressure, I'm doing some horrible drawing here, let's clear everything. The pressure of my pleural cavity in this situation is equal to the pressure in my lungs. So there's no chance for that air to do that thing where it tries to leave the lung, and instead, what happens to the lung? It collapses. So you always have to have a lower pulmonary cavity pressure. Always have to have a lower pulmonary cavity pressure. And that's that difference between the pleural cavity and the lungs is called your transpulmonary pressure. And this is what happens when it equals 0. We don't want it to equal 0. So to reiterate, we always got to have pleural cavity pressure lower than lung pressure. That keeps the lungs from collapsing. And you should know these numbers, okay? All right. Another thing we got to be aware of is this stuff called Boyle's Law. What Boyle's Law tells me is that pressure and volume is inversely related. So in my container here, I got gas molecules bouncing off the walls, exerting pressure. If I lower this part and make my volume smaller, my volume decreased. Because my volume decreased, the little gas molecules are going to hit the walls more often, and that means my pressure is going to go up. So pressure and volume are inversely related. This is key. So what happens when we're breathing quietly? Well, in order for air to move in, we need intrapulmonary pressure to be lower than atmospheric pressure. We're going to achieve this. We're going to decrease intrapulmonary pressure by increasing lung volume. We increase lung volume by increasing thoracic volume. Remember that pleural fluid? We increase thoracic volume by contracting skeletal muscles. So think about it. Your diaphragm contracts, your thorax gets bigger, your lungs get bigger. Because your lungs get bigger, their pressure goes down. It's now lower than atmospheric, and air rushes in. That is our quiet inspiratory sequence. Now, in this situation here, atmospheric pressure is 760. Intrapulmonary is 760. So they're equal. So no air is going in, no air is going out. And of course, my pleural cavity pressure, 756, less than that 760. That keeps my lungs from collapsing. Now notice the diaphragm here is relaxed, curved upward. When the diaphragm contracts, it goes down. So thoracic volume and lung volume increased, which means lung pressure went down. Lung pressure is now 759. Atmospheric is 760. So air is going to go in. Notice that when my diaphragm contracted, my pleural cavity pressure got lower as well, but it's still less than intrapulmonary, so the lungs don't collapse. All right, cool. The major muscle of quiet inspiration is this big muscle here, the diaphragm. In its relaxed state, it is curved upward. It flattens out when it contracts. That pulls the lung down, both sides, increases lung volume, decreases lung pressure. The diaphragm is your major muscle of inspiration. If you're sitting here quietly right now listening to me, and you're just breathing, as you breathe in, you should feel the wall of your stomach, like your abdominal wall, go out as you breathe in. Because your diaphragm is coming down and squishing your guts. If you're using your diaphragm, then your abdominal wall should go out as you're breathing in. Okay, another muscle of quiet inspiration are these little guys between your ribs called the external intercostals. What they do is pull your ribs up and out like a bucket handle motion, and that increases the lateral dimension. It makes the lungs wider, basically. And again, if you're getting wider or taller, your volume's going up, which means pressure's going down. Now, the intercostal muscles aren't as big or as powerful as the diaphragm, which is why we want to use our diaphragm when we are breathing. Don't be a rib breather, be a diaphragm breather. Okay. These muscles are controlled by nerves. The external intercostal muscles, no surprise, controlled by the intercostal nerves. The diaphragm is controlled by the phrenic nerve, which you might have learned about in A&P1, as it comes out of the cervical plexus up in your neck. All right, what if I want to take a big breath in? If I want to take a big breath in, well, I need an even bigger pressure gradient. So I need my lung pressure to be even lower, which means I need my lung volume to be even bigger. So my thoracic volume has to get even bigger. Well, how do I do this? I'm going to do it, number one, by using more motor units. So I'm going to use more motor units, more muscle fibers in my diaphragm and my intercostal muscles, my external intercostals. I can contract my diaphragm harder, like more vigorously, to move it more to make my lungs even bigger. Same thing with my ribs. Like, think about it. Take that big deep breath in like I just did and feel your ribs flare out. So you're using your intercostal muscles, your externals, even more. Then what I can do is use other muscles. I can use muscles like my pectoralis minor. Notice how the pectoralis minor is going to pull up on those ribs. I can use muscles like this guy. That guy is my, come on, let me draw. There we go. Let me draw, let me write. My sternocleidomastoid, which is going to pull my sternum and ribs up. I can also use this group of muscles over here. This group of muscles over here, the long vertical group, are my erector spinae. Do me a favor. If you're sitting down or standing up, all of a sudden, like, make your spine as straight as possible. Like, sit up. You should feel your thoracic cage, like, literally just get bigger. So these are all accessory muscles that can help increase my thoracic volume and thereby increase my lung volume when I want to do a forced inspiration. Okay, what about my quiet expiration? Well, my phrenic and internocostal nerves will stop firing. My diaphragm and my external intercostals will relax. When they relax, my diaphragm goes up. My ribs go in. Thoracic volume is going to drop, meaning lung volume is going to drop, meaning lung pressure is going to go up, meaning air is going to go out. So this is my basic quiet expiratory process. Notice that there's just relaxation. There's no muscle contraction. And I don't need contraction because don't forget, what are your lungs covered in? They're covered in elastic fibers, which you stretched when you breathed in. So they're recoiling right now when you breathe out. All right, and this dude, atmospheric, and intrapulmonary pressure the same, so no air is moving. Notice his diaphragm is currently contracted. When it relaxes, it goes up. The volume in your lungs decreases. Thereby the pressure increases. Now lung pressure, intrapulmonary, is greater than atmospheric, so air goes out. And of course, intrapleural pressure always stays below intrapulmonary, so there is no collapsing of the lungs. Fantastic. Now, what about breathing out? Like, you want to blow out a candle, you want to blow up a balloon, you want to breathe out more. Well, that's called forced expiration. The pressure gradient is going to need to be even bigger in this case. So lung pressure will need to be even bigger, which means lung volume needs to get even smaller, which means thoracic volume needs to get even smaller as well. And to do this, we use muscles, particularly our abdominal muscles. So our rectus abdominis, our transversus abdominis, our internal and external obliques. All of those are going to squeeze your abdominal cavity. And if you squeeze your abdominal cavity, you're squeezing your guts, your intestines, and they're going to be pushed upward. And what are they going to run into? They're going to run into the diaphragm. And they push the diaphragm up, which is going to then make your thorax and your lungs smaller and raise the pressure. Now, another muscle you can use is your internal intercostal muscles. We got to stick the word internal in here. I don't know why we don't have the word internal. It's the internal intercostals. They're going to move the ribs inward, and if your ribs go inward, your thoracic and lung volume is going to decrease, making lung pressure increase. So that is how you do some forced expiration. You use these extra muscles. And with that, folks, we are done our pressure talk and our mechanics of breathing. I want to switch to a new topic right now, and that new topic is airway resistance. Now, we made a big deal about peripheral resistance for blood flow. Ain't such a big deal for air. So airway resistance is normally insignificant because airways are relatively wide and air is not thick. Air has a low viscosity. Now, if you are having an allergic reaction, peanuts, bee sting, whatever, or if you're having an asthmatic reaction where you either get too much muscle contraction or mucus production, well, that can significantly increase the resistance in the airway and make it hard to breathe. Now, in a situation like that, medicines like, an inhaler or an EpiPen can act to bronchodilate, to relax the smooth muscle and get that airway wide. A wide airway, remember, if radius goes up, resistance goes down. All right. Good. So that's resistance. There's another thing we got to consider, though. My alveoli, which remember are my little tiny circular microscopic lung structures where gas exchange happens. My alveoli are lined by water molecules. Water molecules love water molecules. So the water molecules lining an alveolus, they actually want to coalesce in the middle. And that's called surface tension. And surface tension of water is going to want to make the alveoli collapse. Water really likes other water molecules. That's why this water strider can walk on water here. So, do we want our alveoli to collapse? No. If they collapse, we have to uncollapse them with every breath, and that would be really tough. So we need something to fight this tendency of the water molecules, their surface tension, which makes them want to group together and collapse the alveolus. Now, first of all, I gotta have water molecules because I need my my alveolar lining to be moist. I gotta have these water molecules here. What I do is, if you recall, in an alveolus, you have a type 1 alveolar cell that is simple squamous, alveolar macrophage, then also a type 2 cell. We mentioned that a little bit ago. The type 2 cell makes a chemical called surfactant. And surfactant, it is this chemical, I think it might be called like 1,2-dipalmitol-lecithin, but that's not on your test. This chemical is going to reduce the surface tension. So here's the water here lining the alveolus, and the water has this tendency to want to go to the middle, but the chemical surfactant released by these type 2 cells reduces the surface tension. And so that prevents the alveoli from collapsing. It's really important that we have these cells. Now, premature babies, may not make enough surfactant. So in a premature baby, the surface tension can cause the alveoli to collapse. And then it's really hard for the baby to breathe in because every time they breathe in, they got to uncollapse these wet alveoli. Imagine like making a water balloon, then emptying it, and then trying to blow it up with air. If you did that, it would be really hard to do. And that's what the babies have to do, like with these microscopic balloons here. Luckily, there are synthetic surfactants. There's positive pressure breathing devices that can be used. So there's ways around this, which is good for tiny, beautiful babies. All right, done with surface tension. Another thing about the lungs that we have to consider is the lungs' compliance. Compliance is stretchiness. We got to make sure the lungs are stretchy enough to expand, but not so stretchy that they don't recoil. We also need thoracic compliance, like our rib cage needs some stretchiness to be able to expand. So compliance is important. Of course, compliance would be lower if your alveoli would be collapsed. It would be harder to stretch the lungs. Compliance gets lower as you get older, especially because your cartilage gets stiffer and your musculature gets stiffer in here. All right, so that's compliance. And our next topic is we're going to talk about gases. And what I want to do first of all is look at the atmosphere we have here on Earth. The primary gas in our atmosphere is nitrogen. Almost 79% of our air is nitrogen. Almost 21% is oxygen. And we got water vapor, a little tiny bit of CO2, and tiny amounts of other gases that we'll kind of ignore. But these are the gases in the atmosphere. Now what I want you to do is notice the gas composition of our lung air. Notice that oxygen has gone down. Well, that makes sense because the blood is stealing oxygen from the air. CO2 has gone up. That makes sense too because the blood has added CO2 to the air. Water has gone up, water vapor, because remember, one of the things we do in our conducting zone is we humidify the air. Nitrogen has changed too, but that's more of a product of the other changes. Nitrogen is an inert gas. You guys remember you took chemistry, right? Nitrogen gas, N2, has this nice triple bond here. Triple covalent bond, so it's not going to react with anything. Nitrogen is also very insoluble in water, so we don't really care about nitrogen unless we're scuba diving. For scuba diving, nitrogen matters. But if we're not scuba diving, we don't care. Okay, cool. Now, what I want to talk about now is the exchange of gases between the air in your lung and the blood in your pulmonary capillaries. And there's three things we're going to discuss. First of all, there's ventilation-perfusion coupling. Then there's the thickness of our respiratory membrane. Then we're going to talk about partial pressure gradients and solubilities. Now, ventilation-perfusion coupling is a fancy way of saying you're going to match, that's what coupling means, you're going to match airflow, that's ventilation, to blood flow, that's perfusion. So what that means is I got to make sure everywhere in my lungs air is going, I got blood going there too. If there's a region of my lungs that's getting less airflow, it would be silly for me to send blood there. So I'm going to vasoconstrict the pulmonary arteries there. That is an example of how I do ventilation-perfusion coupling. I'm going to match my airflow and my blood flow. Similarly, if there's decreased blood flow to a region, well, I don't want to send air there then. I'm going to bronchoconstrict in that region. So I'm going to match airflow and blood flow. That way I can get good exchange. Now, another thing that impacts exchange is that respiratory membrane. Recall that the respiratory membrane is what's separating the air in your alveoli from the plasma in your pulmonary capillaries. It is going to be your simple squamous epithelium of your capillary, your simple squamous epithelium of your type 1 alveolar cell, and a little bit of protein in between them. This trio is called your respiratory membrane. It can be as skinny as 0.1 micrometers. That's 1/10 millionth of a meter. Imagine taking a meter stick, like you might have in a science lab, taking a meter stick, chopping up into 10 million pieces. Do that for me. That's how skinny this respiratory membrane can be. Now, if this gets too thick because of buildup of fluid, like if somebody's got pneumonia, well, that means you're going to have a thicker exchange surface, and exchange is going to be challenged. Okay, the third thing that matters, and this is really important for exchange, are partial pressure gradients. And the term partial pressure means I can talk just about the pressure of oxygen or just about the pressure of CO2. For example, my alveolar air will typically have a PO2, a partial pressure of oxygen, of 104 millimeters of mercury, and a PCO2, partial pressure of CO2, of 40 millimeters of mercury. Compare that to the blood entering the pulmonary capillary. It has a PO2 of 40. 104, a lot bigger than 40, so air is going to flow from the air into the blood. Meanwhile, the PCO2 of the blood coming in is 45. PCO2 of your air is 40, so CO2 diffuses that way out. So these partial pressure gradients are crucial for exchange. Now, you guys are smart, I know that, and you probably noticed that 104 to 40, that's a big difference, right? 45 to 40 is a small difference. And the reason for this difference is CO2 is a lot more soluble in liquid. CO2, carbon dioxide, is more liquid soluble, so it takes less of a pressure gradient to move it in or out of a liquid. Oxygen's not that soluble, so it takes a bigger gradient to push oxygen into your blood plasma. Okie dokie. We can also do this partial pressure gradient analysis in our tissues where we're doing systemic gas exchange. So the blood coming into a systemic capillary has a PO2 of 95. Inside my tissues, the PO2 is going to be 40 or less. So oxygen is going to move from the blood into the tissues. Meanwhile, inside my tissues, the PCO2 is about 45 or more, whereas the blood coming into my tissues has a PCO2 of 40. So CO2 is going to move from the tissues to the blood. Fantastic. And again, big gradient for oxygen because it's insoluble, or let's say less soluble, and smaller gradient for CO2 because it's more soluble. And with that, we can keep on going, trying to wrap up here, and talk about oxygen transport. Now, oxygen is transported primarily, 98.5% of it, attached to hemoglobin. Here is a hemoglobin protein. We know we have millions of these in our red blood cells. Each hemoglobin protein has 4 little heme groups. Each heme group has an iron attached. The iron is what actually grabs onto the hemoglobin. So 98.5% of your oxygen is attached to the iron in hemoglobin. The other 1.5% is dissolved in plasma. Remember, we just said oxygen is not that soluble. Now, hemoglobin with oxygen attached is called oxyhemoglobin. Hemoglobin without oxygen is called deoxyhemoglobin or reduced hemoglobin. Now, every hemoglobin, because it has 4 heme groups, 4 iron molecules, can carry 4 oxygen molecules. Here we got a nice little chemical equation. This is reduced or deoxyhemoglobin over here. Deoxyhemoglobin plus oxygen gives me oxyhemoglobin plus a hydrogen. And notice this reaction can go in both directions because hemoglobin can pick up oxygen, but it also can let go of it. In the lungs, where we have high oxygen levels, this reaction runs to the right. We make oxyhemoglobin in the lungs because we're picking up oxygen. In our tissues, where oxygen levels are lower, the reaction goes to the left. So oxyhemoglobin lets go of oxygen, leaving us with deoxyhemoglobin. So we're letting go, we're dropping off oxygen in our tissues. Now, CO2, heat, and low pH all cause this leftward reaction to occur, which makes sense because what do you make in working muscles? CO2, heat, and acid. And so all these products of working muscles, working tissues, push this reaction to the left, meaning hemoglobin is letting go of oxygen in working tissues. There is a concept called hemoglobin saturation, and if we look at lung blood is almost 100% saturated. That means every hemoglobin, like 4 out of 4 spots for oxygen are filled. Similarly, in our systemic arteries, we're practically 100% saturated. Again, 4 out of 4 spots on every hemoglobin for oxygen are filled. Now, what are you doing right now? Are you just sitting down, listening to this excitedly, intently? You're probably like not exercising as you listen to this right now. I'm going to assume you're not working out while you listen to this. And if you're just at rest, your veins, surprisingly, are 75% saturated. That means in your veins, 3 out of every 4 spots are occupied by oxygen. 3 out of every hemoglobin spots are occupied. This is surprising because we were always like, veins are blue. Veins are deoxygenated. Yeah, it's not exactly true. Veins are just less oxygenated. And this fact that veins still have 75% oxygen is called the venous reserve. Because if you needed it, if you got up and started sprinting away from your desk right now, you sprint out of your room, you sprint down your first flight of stairs, down your second flight of stairs, you sprint out of your door, you sprint down your driveway, you sprint out the gate. It's a great house. You would need that extra oxygen, and that oxygen is called, that extra oxygen at 75% is called your venous reserve. Fantastic. I'm going to kind of ignore the graph. I know that makes you sad, and I apologize that you're sad about ignoring the graph. I'm sorry. I'll make it up to you. Okay. What about CO2? CO2 transport. Well, remember, CO2 is more soluble than oxygen, so 10% dissolved. Remember, we only had 1.5% oxygen dissolved in plasma, 10% CO2. Another 20% of your CO2 is attached to hemoglobin, bound to the amino acid part of hemoglobin, and hemoglobin with CO2 attached is called carbaminohemoglobin. The other 70% of your CO2 is going to exist as part of bicarbonate. Remember, CO2 adds with water, and that is going to give us, well, first it'll give us an intermediate called carbonic acid, which is H2CO3. And then H2CO3 is going to dissociate and give us HCO3 minus and H plus. And that HCO3 minus right there, that is bicarbonate. So most of your CO2, 70% of it, exists as bicarbonate in your plasma. Okie dokie. Let's look at a picture of all that. Get that out of here. So, tissue cells make CO2. CO2 diffuses from them down its pressure gradient, into the interstitial fluid, into the plasma. 10% of your CO2 dissolves in the plasma. The rest of your CO2, almost all of it, we'll cover this a little bit in a second, almost all the rest goes into your red blood cells. In your red blood cells, 20% of your CO2 combines with hemoglobin to form carbaminohemoglobin. The other 70% of your CO2 inside the red blood cell combines with water to form carbonic acid, which then splits to form bicarbonate and a hydrogen. Now, this reaction happens primarily in the red blood cell. You may correctly wonder, why does this reaction not happen a great deal in your plasma? The plasma is 90% water. Well, the thing is, the enzyme that catalyzes this reaction, it's known as carbonic anhydrase, and it is found in your red blood cell. It is not found in your plasma. So this reaction preferentially happens in your red blood cell. So you make the bicarbonate in your red blood cell because the enzyme is in there. However, the bicarbonate quickly leaves the red blood cell and goes into the plasma, and it hangs out there. Because the bicarbonate leaves, a chloride ion actually goes in to make sure we stay like neutrally charged. Negative out, negative in. So this is what happens in our tissues. If we switch to the lungs, the opposite happens. Bicarbonate goes in the red blood cell, chloride goes out. Bicarbonate combines with hydrogen to make carbonic acid, which then splits to give me water and CO2. CO2 diffuses into my lungs air, or my alveolar air. This reaction happens a great deal, like 1 billion times more in the red blood cell than in the plasma, because we got the enzyme carbonic anhydrase. Meanwhile, that was 70%. 20% of your CO2 was part of carbaminohemoglobin. Hemoglobin lets go of CO2. Then the CO2 diffuses. The dissolved CO2, of course, will diffuse as well. And hey, we can see what oxygen is doing here as well on both pages. All right. Wonderful. We're almost done, folks. This is great. Getting so much accomplished. What I want to talk about now is your control of respiration. So most of your respiratory control, whoops daisy, is going to happen in your brainstem, in your medulla oblongata, and in your pons. So here's your pons right here. In your pons, you have a pontine respiratory group. It's a group of nuclei in the pons. In your medulla oblongata, you have a pair of respiratory groups, clusters of neurons. One in the back, called the dorsal respiratory group. One in the back, one in the front, called the ventral respiratory group. Now, of these, the mostest, importantest, is the ventral respiratory group. It is the head one. It is the one truly in charge. Let's deal with the minor ones first. The dorsal respiratory group helps your respiratory system integrate sensory information, like what's your blood pressure, what's your body temperature, what are your muscles doing right now. All of those things can impact respiratory rate and depth. The pontine group modifies breathing during sleep, exercise, when you have big changes in the rhythm and depth of your respiration. Now the ventral respiratory group, though, it is the primary control center. So there it is, cluster of neurons in the anterior medulla oblongata. It has in that cluster inspiratory neurons that stimulate the phrenic intercostal nerves. Remember, the phrenic nerve goes to the diaphragm. Intercostal nerves go to the intercostal muscles. So the inspiratory neurons stimulate those nerves, those muscles contract, and we breathe in. Expiratory neurons, on the other hand, inhibit the inspiratory neurons, and then we breathe out. So like 3 seconds where the inspiratory neurons are turned on, then 2 seconds where the expiratory neurons are turned on, roughly. The biggest thing influencing the VRG is the pH of your CSF, cerebrospinal fluid. Minor influences include plasma pH, plasma PCO2, and plasma O2, PO2. So, on your ventral respiratory group, you have special neurons called central chemoreceptors. So central chemoreceptors are special neurons. They're literally on the ventral medulla. They measure your CSF pH, and they stimulate the ventral respiratory group. They stimulate those inspiratory neurons if pH goes down, because we don't want CSF to get too acidic. That is bad for my brain tissue. So, CSF pH depends on CSF PCO2. Remember, CO2, what does CO2 react with? CO2 reacts with water. And when CO2 reacts with water, I got bicarbonate, but I also got a hydrogen ion. So if I have more CO2 in my CSF, it gets more acidic. I can get more CO2 in my CSF if I get more CO2 in my blood. So if you hold your breath, this plasma PCO2 will go up, making CSF PCO2 go up, making CSF pH go down, which is going to then make you want to breathe. And it's going to force you to breathe eventually. Okay. Fantastic. Whoops. Wasting time here. So, let's say you held your breath. Your PCO2 of your plasma would increase because you're holding your breath. You're not getting rid of CO2. What would that do to CSF PCO2? Well, if there's more in your blood, the amount in your cerebrospinal fluid is going to, I'm hoping you just shouted out increase at the top of your lungs. You like slammed on your table and said increase. And if we have more CO2, what happens to hydrogen levels? Don't forget, CO2 plus what? Plus water. CO2 plus water. It gives us bicarbonate plus a hydrogen ion. And that hydrogen ion means if I got more CO2, I get more of it. So this goes up. meaning pH goes down. And our brain doesn't like this, so our brain says, no, we got to get rid of CO2. So our respiratory rate and depth are going to go up. And this is the main way you regulate respiration, is this sequence here. Now, I did say there are other things that influence your VRG. So you have other chemoreceptors called peripheral chemoreceptors. They're in little areas called your aortic bodies and carotid bodies, little neuronal regions in your aorta, in the arch there, and in your carotid sinus here. And these guys are activated by an increase in plasma PCO2, or a decrease in plasma PO2, or a decrease in plasma pH, but they're a lot less sensitive. We're more tolerant of changes in our blood, less tolerant of changes in our CSF. Other things influencing respiratory rate include body temperature, so direct relationship there. Blood pressure, same idea. Plus, of course, your brain. I mean, think about, like, you can hold your breath. You can voluntarily breathe faster, breathe slower if you want to. If you get excited, if you're crying, that affects breathing rate as well. Irritants. If you get pepper sprayed, it's going to mess with your breathing rate. Definitely. All right, almost done. Wrapping up. What would happen if you were holding your breath, if you were hypoventilating? Well, if I hypoventilate, I'm retaining CO2. And we already established if there's more CO2, there's more acid. And if there's more acid, pH is going to go down. And that's called respiratory acidosis. Similarly, if you hyperventilate, you're getting rid of CO2. So less CO2, and that means pH is going to go up. You got less acid, so pH goes up. And that is known as respiratory alkalosis. All right, final 2 slides of this lecture. Coming in at less than an hour and 15 minutes, I'm wagering. This is really good. There is something called metabolic acidosis. That is a drop in plasma pH for non-respiratory reasons. Like, for example, if you drink way too much alcohol, alcohol is going to have acidic products that you make from it when you metabolize in your body. So more alcohol, more acid, that's called metabolic acidosis. If you have kidney disease, that is a kidney right there. So if you have kidney disease, we have alcohol can cause metabolic acidosis. Kidney disease is going to cause metabolic acidosis because the urine is acidic. So if your kidneys are not working, you don't make urine, and you don't pee away that acid. You retain that acid, you become more acidic. If you're starving, then you are going to break down muscle and fat. And when you break down muscle and fat, you are going to, that says starvation. It doesn't say stewation. It says starvation. If you break down muscle and fat, you make acids from that. Similarly, if somebody has untreated diabetes, you start breaking down muscle and fat from untreated diabetes, and then it can also cause metabolic acidosis. These are prunes over here. If you eat a whole bag of prunes, you are going to be having some liquefied poop. And when you're pooping excessively, well, poop is actually basic. And so if you're pooping away a lot of base, you become more acidic, and that's metabolic acidosis. Now, in this situation, what would your respiratory system do to compensate? So if you became more acidic, you would actually increase your breathing rate. You would increase your breathing rate to get rid of CO2 and thereby get rid of acid. Okay. One more slide, and that's about metabolic alkalosis. This is a rise in plasma pH for non-respiratory reasons. We just said poop was we just said poop was basic. So if you're retaining poop because you're constipated, if you're retaining poop because of constipation, that can make you more alkaline. If you overdose on antacids, that can also make your blood more alkaline. Or if you lose too much acid because you're vomiting, then you can, it's more vomit-like color, then you can become more acidic. Sorry, more basic if you lose too much acid. Okay. How would the respiratory system compensate? Well, we would compensate by hypoventilating in order to retain CO2 and thereby retain acid. And with that, my friends, we are totally done this respiratory system, this respiratory lecture. Check in with me if you have any questions. Let me know. Bye.